Associate Professor, Department of Biological System Engineering, WSU. On the behalf of CAS project of Mahatma Phule Krishi Vidyapith Rahuri, I extend warm welcome to you. I also extend warm welcome to Dr. Sunil Gorantiwar sir, co-convener of this training program and principal investigator of CAS project. I also welcome Dr. Mukund Shinde sir and Dr. Sachin Nalode sir, organizing secretary of this training program. Now I would like to request Dr. Mukund Shinde sir, organizing secretary of this training program, to introduce Dr. Sindhuja Sankaran ma'am to our participants.
Yeah, thanks, Dr. Deshmukh. And hi and good morning, Sindhuja from India. So welcome all the participants, all the dignitaries. It is my privilege to introduce Dr. Sindhuja to all the participants. Dr. Sindhuja is a researcher in agricultural automation engineering. She engages in sensing data-driven discoveries that transform how we study the interaction between crops, genetics, and environment to produce sustainable food, fiber, and fuels. She is based at Washington State University in Pullman, Washington. Her work focuses on advanced sensor technologies that detect and measure phenotypes, the physical expression of genes in crops, supporting plant breeding, crop plant research, and precision ag. She utilizes optoelectronic, biological, and chemical sensors for non-invasive, rapid, and continuous monitoring of plant responses to environmental stress. She is helping to create a faster and better understanding of how our food crops react to changing environment. Over the past 6 years, she has developed new tools to evaluate traits, including crop vigor, stress tolerance, and seed size and quality across multiple crop breeding programs. She is a member of the American Society of Agricultural and Biological Engineers, the National Association of Plant Breeders, the North American Plant Phenotyping Network, and the International Society of Precision Agriculture. Ma'am, I welcome you on behalf of Center for Advanced Agricultural Science and Technology for Climate Smart Agriculture and Water Management, Mahatma Phule Krishi Vidyapith Rahuri. Thank you.
Thank you very much, Dr. Shinde, the organizing secretary, for introducing Dr. Sindhuja Sankaran ma'am. So without wasting much more time, I would like to request Dr. Sindhuja Sankaran ma'am, Associate Professor, Department of Biological System Engineering, WSU, to start your lecture and presentation.
Thank you for the kind invitation. I would like to share my slides. So I'm just... Just give me 1 second. Are you able to see the screen?
No, ma'am.
Okay. Now?
Yes, yes. It is visible.
Okay. So I would like to start my lecture. Before I begin my lecture, I would like to thank the organizers for giving me this opportunity to present about sensing technology for phenotyping applications. I'm going to explain what phenotyping is in few minutes and give a broader perspective of it. So as Dr. Shinde introduced, I'm from Washington State University. I'm an associate professor here. I have been here for 7 years. Before that, I have been in other universities, University of Florida, as well as North Dakota State University and Iowa State University. I'm a part of agricultural automation engineering, which is a focus group within the Department of Biological Systems Engineering. And I know earlier during this event, you have heard from Dr. Lavkod as well as Dr. Chongyuan Zhang, my colleagues from the department. So all of us work towards different focus areas within ag automation systems. And my focus is basically sensing applications for phenomics. Okay, so all our research is tied to food security. We know that the global population is going to reach 9 billion by mid of this century, and we are worried that we would not have enough food to feed for all. This is not just because of population increase, but we know agricultural land is also becoming smaller. So there are other stress factors influencing these conditions, including climate change. So throughout the world, there are different breeding and genomics programs that are focused towards new crop variety development that can produce more food with less resources. So quite often, it is thought that food security is associated with a growing population increase in food demand, but it's not just the food security, but it also involves other aspects. One aspect is food safety. You want to produce food that is safe, is rich in vitamins and other minerals. At the same time, consumers are becoming very specific in their needs. So they, even if you are looking for tomatoes, there may be different types of tomatoes, and each of us may have a preference. In US, we have a lot of types of apples as well. And again, based on each person, the preference is going to be different. And also the final aspect is sustainability. We know that there is climate change. We know that the water resources are going to get limited. We want to produce more crops or more food with less nutrients, less chemicals in the environment, less water for sustainable production. And the way to achieve any of this is through developing new crop varieties, not just maintaining and managing the current crop varieties. So that's why breeders and scientists are involved in the process of developing new crops by changing their genetics. In Washington state itself, we have a number of breeding programs. We have at least 15 different types of breeding programs, which involves cereal grains, which is wheat, barley, buckwheat, and quinoa. We have legumes or pulses, chickpea, pea, lentils. Many of these are also grown in India, as well as we have a lot of horticultural crops, which includes potato, apples, pear, peach, cherry as well. So my research focus is I closely work with these breeding programs to help them use sensing systems for better decision making. So just going back to the basics on population, similar as many of you may have siblings, and each of the sibling is very different from us. Right? So if two parents are going to have 10 children, the 10 children are not going to be the same. The same way, when you have crop varieties and you mix two different varieties, it's going to produce 100 different types of progenies that are going to have its own characteristics. So once they have this 100 different varieties or progenies or lines or entries, whatever they may call it, you need to phenotype. So phenotype is evaluating the plant responses to the environment, as well as also evaluating the plants for trait of interest. For example, if they are developing disease-resistant varieties, they are going to inoculate all of these 100 different varieties with a certain disease and see which variety is trying to resist it much better. So that aspect is called as phenotypic description, and quite often, sometimes it involves manual measurements, sometimes it involves visual rating. So it can be subjective, expensive, as well as laborious. When it comes to genotype, there is also genomic technology where you try to identify the genetic makeup. So the genetic makeup is, it's just like we can take a hair sample and tell what is our genetic makeup, right? The DNA sampling or DNA fingerprinting. But phenotype is a little more challenging than the genotyping itself, because if I may have a rice variety or a wheat variety that I grow in Washington state, and the same wheat variety, though it's genetically identical, it's going to behave differently when I grow it in India. So phenotype is a dynamic characteristics which is a function of not just the genotype but also the environment. And mainly we are trying to identify phenotype because we want to identify genes that is associated with a certain traits. So all this I'm probably oversimplifying this, but I wanted all of you to understand the meaning of phenotype and genotype before we proceed further. Okay, so again, this is the same concept I mentioned. So genotype interacts with the environment, and that results in a phenotype, right? And on an average, for any variety to develop, it takes around 12 to 20 years. For example, for new beet variety to grow develop, it takes at least 10 to 12 years. But when it comes to tree fruit species like apples, it may take up to 20 years because it takes at least 3 years for the planting body to fruit. So the genetic development or the genotype development or variety development or crop improvement is a very long process. Okay, and phenotyping is challenging because it is dynamic in nature. In addition to that, I mentioned that it can be very laborious. So for example, if I have thousands of different breeding lines, it's going to take a very long time to evaluate each of those if we do not have a rapid method. It can be expensive. Generally, breeders hire a lot of personnel to go to the field conditions and evaluate their traits. And it can be subjective. For example, it can be disease rating, which is visually assessed, looking at the symptoms. They say, okay, this symptom is mild, severe. They usually rank it from 1 to 10 or 1 to 5. They measure plant height using yardstick. They measure visually when it is flowering as well as emergence. So it can be very subjective by nature. So when we use the sensing technique, it can help in many different ways. It can make it more accurate. For example, if someone is evaluating the disease symptom, it is limited to visible band as well as there may be perspective. So I may say this is mild while my colleague may say this is moderate symptom. So there is subjectivity with it. When we use sensing techniques, it eliminates those subjectivity. It can be high throughput, it can be rapid, it can be non-destructive, it can be automated, it can be less expensive, and sometimes some of these sensing techniques are able to see beyond the visible range. So those are all the benefits of using sensing techniques, and that's why the term phenomics is nothing but phenotype in using sensing, automation, and data mining methods, where you can capture more information on those traits rather than the standard method. Okay, so in our lab, we use a range of different sensing tools. We use handheld sensors, tractor-based systems. So handheld sensors can give you more information than a standard eye can using sophisticated instrumentation. It can be a tractor based. On the top, the tractor based system is used for field crops, while in the bottom it is used for tree fruit species, as well as unmanned aerial system based sensing. So all this are possible. In addition to that, more recently, we are able to use the satellite imagery to phenotype as well. So I did not use any examples in this lecture, but if you refer to our recent work, we have published on satellite-based imagery for phenotyping applications as well. So in addition to the platforms or systems itself, we use a range of sensors. We use a multispectral sensors, and I think some of my colleagues may have explained it. So instead of just red, green, and blue bands, they have more information on it. So they have near infrared range as well. In addition to that, we have hyperspectral sensor, which has hundreds of bands. We have LiDAR-based systems, X-ray, CT we use, RGB and thermal camera as well. So, and in our lab, we have used many of these sensing techniques to evaluate a range of traits in a range of crops. For example, wheat, we have used it to evaluate emergence, yield potential, potato, water use efficiency, apple is disease as well as architectural. So we have used a range of sensors to evaluate many different traits. And today I'm just going to give you an overview of some of those techniques, just a brief outlook on examples of some of these applications. Okay, I would like to mention quite often phenomics is, or phenotyping using sensing systems is done under both controlled environment and field environment. And field environment is much more challenging than the controlled environment, basically because the environment in field conditions is hard to control. So even so, with sensing techniques, you get good correlations with the trait of interest using sensing. So when you look at the electromagnetic spectra, all our eyes can see is the small range of visible band, right? So we are only able to utilize this, I'm sorry about that, to see that small band of visible, but there are sensors that are capable of seeing beyond this visible range. So there are sensors that are capable of seeing infrared, microwave, radar-based technologies. We have X-ray, CT, gamma-based sensing systems. So there are sensing systems that utilize beyond the visible range for detection as well. Okay, and many of these systems can not only monitor the crop responses, it can also monitor the produce or product quality. So crop responses is, okay, plant height, disease symptoms. So those are crop level traits that we are interested in. In terms of produce and product quality, I'm thinking about seed size, seed color, or apple color, all those traits of interest that is associated with the final product or produce. So examples again of crop responses: water use efficiency, nutrient use efficiency, growth factors, crop losses, crop diseases, and pests. When it comes to produce or product quality, it is tuber, for example, potato tuber, color, shape, quality, starch and sugar content, storability, sprouting, post-harvest losses, as well as product quality as well. Okay. From now on, I'm just going to talk some of the examples on using some of these technologies. So my first example, this is on potato. So most of my examples are associated with breeding programs. So I closely work with a number of breeders. So this was Dr. Sagar Satwalli. He's a potato breeder in Oregon State University. And we were evaluating potato virus Y, which is a type of virus disease. And we were evaluating the resistance to this disease. And we could see that just using a multispectral imaging, we can monitor the rate of growth of the plants, and clearly using, you can also use RGB imaging, but multispectral imaging, it's easier to eliminate the background from the crops itself. And with the rate of growth, so if you really see canopy vigor in terms of number of pixels per plot, we could find that it is closely related to the disease resistance as well as yield. So this method not only worked for potato virus Y, it also worked for late blight as well. So this is one of the examples. Rather than a breeder going and evaluating and looking for visual symptoms, you can quantify the phenotypic responses in a better way using remote sensing technologies. So the second example is associated with crop growth factors. So this is in terms of emergence. So we have already published, and the citation is given below here. It is published in American Journal of Potato Research. And you can see that in early stages of potato growth, we had the breeder go and count manually the emergence of the potato plants. They were evaluating the effect of growth hormones to control the emergence of potato plants. But some of those traits, not only number of plants, it also canopy closure, all those factors can be measured using remote sensing tools. So in this research, we also used multispectral imaging to be able to evaluate the potato emergence by counting the number of potato plants that are coming. Another example on crop growth factors is the emergence of winter wheat itself. One of the issues associated with winter wheat is, since winter wheat is planted before the winter season, sometimes if the weather is too cold and the soil crust is too hard, it's hard for the plants to emerge. So what a breeder usually, this is a winter wheat breeder, Dr. Aaron Carter, from Washington State University. The way they evaluate the emergence is by looking at, going and evaluating and rating them from 0 to 100 at every 10. So they rate, 0, 10, 20, depending on the rate of emergence. Instead of that, we used a remote sensing technique, and you can clearly see, especially the image in the right, that some of those varieties that have emerged well, you can better quantify using a remote sensing data. And we did compare it with the standard measurement, which is the visual rating. And you can see over here, it's either 10, 20, 30, 40, that's the way it's evaluated. But with image data, you can get a continuous number, and it was very highly correlated with the visual rating by itself. And again, this is one of our published work from past. So again, not only the visual, not only the emergence, you can also evaluate the yield potential as well. And this, the image in the bottom with colored box, it's just looking at the field and seeing, which varieties are evaluated at a higher rate. And I should also mention that you can also study the spatial variability within your field using remote sensing data. So that is the major advantage. When you are going and rating your plots individually, you cannot observe the spatial variability in the field. But as you're able to go and get this data and look it visually, visualization becomes very easy and important, and you can make a better judgment of your field conditions as well. Okay, so those were a couple examples of emergence, crop growth factors, as well as diseases. Other studies include water use efficiency. In this study, we were evaluating 9 different potato varieties, and they were having 2 irrigated conditions, and there were multiple seeds, multiple replicates, and you can see that some of these vegetation indices that were extracted using proximal sensing data. So this was a hyperspectral data. where we use the near infrared and some of the specific wavelengths, we could see clear distinction between the varieties. So 1 to 9 is the variety itself, and 65 and 100% is 65% ET and 100% ET. So it's different irrigation treatments. And you can see that some of this variety, for example, variety number 4 had a good performance, even in spite of having lower irrigation rate. So there are some varieties that can grow well even with less water, and we are interested in identifying those varieties using sensing techniques as a way to improve the sustainability. Okay. In addition to some these traits, another trait that can be easily evaluated using remote sensing is plant height. Rather than going plot by plot, taking a scale to measure the plant height, you can just use unmanned aerial system or drone-based imagery to reconstruct these 3D plots and be able to measure the plant height. And I think Dr. Chongyuan Zhang will briefly touch base on the process for this as well. So this is another applications of using remote sensing for plant height estimation. I also wanted to move to some of the other traits, which is for crop, in terms of crop produce or quality. I wanted to talk, here is an example of seed size. Traditionally, when they do sieve analysis to measure the chickpea size, so they just have a series of seeds where they shake it with a mechanical shaker to measure the seed size. You can just use a simple imaging system, a simple digital camera to measure the seed size. So we did so, and we got a very high correlation of 0.89 compared to the sieve analysis. Not only can we do that, we can, I should also want to mention that you can not only see the seed size, you can also study the seed size distribution. So it gives you more information than the standard size. The same distribution cannot be, if depending on 5 to 10 seeds, you may get 5 to 10 different seed size categories, but you cannot evaluate or get the distribution. But when you use a simple digital imaging to be able to do a seed size, you can easily get a seed size distribution. So these are benefits, these are advantages of using sensing techniques for seed analysis. Another example is potato size. Potato is one of the 4 to 5 most grown crop throughout the world. Many countries, almost all different regions grow potatoes. So potato is an important food crop that is studied in many different countries. Breeders in Washington are also interested in potatoes. Unfortunately, not as a food crop, but more so for processed food, mainly chip industries. So they are interested in getting potatoes of certain length to width. In general, the length to width is measured using a caliper or a scale. And this is true for any type of tuber breeder. So it's, though this application is for potatoes, but it could be carrots, it could be cassava, it could be any crop of interest. So many people are just using this calipers or scales to measure the shape, size of the tubers. In one of our studies, we are interested in using image-based detection of the produce itself. So you can easily measure the size of tubers or any other vegetables or fruits using imaging systems. Technology is developing so rapidly that we are also trying to develop apps to do the same thing. So there is a lot of potential in some of these technologies. I think this is one of the last examples I wanted to talk in terms of produce quality. And again, I was talking about subjectivity. So bitter pit is this dark spots in apple, and it is a disorder. It's a important disorder. So especially in Washington state, there are certain apple varieties that are prone to bitter pits. And the breeders are trying to develop new varieties that are resistant to bitter pit. And the way they evaluate bitter pit is through visual assessment. And you can see over here, like they are just looking at the symptoms outside. But in one of our study where we used X-ray CT, we found that almost 40 to 66% of the bitter pits are inside the apple rather than outside the apples. So so when they are evaluating the apple varieties for bitter pit resistance, they are almost missing out on 40 to 66%. That's where some of these sensing techniques can bring value to the applications. Okay. With that, I wanted to transition to a different topic. I just want to briefly introduce what is machine learning and explain it with just one example as a part of this lecture. So I told some of these already. We have there are different platforms you can use for sensing application, which could differentiate from towers to tractors to drone, aircraft, and satellite imagery. All these technologies can be used for phenotyping applications. In addition to that, there are many different sensing techniques that can be used. The most commonly used ones are RGB imaging, multispectral imaging, hyperspectral, thermal fluorescence, as well as the 3D sensors, which is like LiDAR and other 3D type of sensors. Okay. When you come to phenotyping applications, we are talking about image acquisition using different types of sensors. But there is a lot of steps involved in the process, and the process can range from image acquisition, pre-processing, segmentation, feature extraction, and classification. So there are a lot of steps, and I think again Dr. Chongyuan Zhang will talk to you about some of those standard steps when you analyze the image to extract the features. But I wanted to mention that though there are this is a standard protocol of data analysis, you can apply machine learning methods to each of these processes. So that's why machine learning techniques are commonly used in phenotyping applications as well. If you really broadly classify the machine learning aspects, it could be, there are different categories. The first one is identification and detection, classification, quantification, as well as prediction. So detection is nothing but absence and presence. For example, if a disease is present or absent. So is my plant having nutrient deficiency or not? So it's just a yes or no type of answer. When it comes to classification, you can categorize in more categories rather than just the yes or no answer. So for example, if nutrient deficient, if it's nutrient deficient, you can specify whether it's nitrogen deficient, phosphorus deficient, or is it a micronutrient deficient. So you can classify it into different categories than just saying yes or no. And the third one is quantification, and the fourth one is prediction, where you're trying to predict what is a specific value. So prediction, a good example for prediction is predicting the yield or predicting the stomatal conductance, transpiration rate, photosynthetic rate. So predicting a certain outcome rather than saying, okay, does it have nutrient deficiency? What type of nutrient deficiency? So it's a more a quantitative output, and machine learning can be used in any of these outcomes. So in machine learning, I should say that there are a lot of pre-processing steps, which I'm not going to go over in detail. And in terms of models or approaches, there are hundreds of different approaches that can be applied for different applications. And nowadays, a more commonly investigated approach is deep learning, or CNN or deep learning is becoming more common, not only in different, not only in agriculture, in all different sectors as well. Okay, so in general, machine learning, what it does is it learns patterns. So it is trying to identify the patterns as well as similarities and dissimilarities in data. It has practical implications, and you may have seen this in a lot of movies, right? Face recognition, or there are people do sophisticated things, at least in movies, but people use it for many of those. For example, in potato chip industry, they just use it to identify a burnt chip and remove it from the processing or packaging line. And there are many different softwares that have some of these tools inbuilt. So it can be OpenCV, it can be MATLAB, it can be Python. There are a lot of tools already inbuilt into any of these programming languages. I think I'm going to skip that aspect. So I'm just going to talk to you about apple breeding program, and I'm just using one study where we used many different methods to evaluate just one trait, disease resistance. But I would like to mention over here. So for example, in apple breeding program, they evaluate many different traits. Disease resistance is one. They evaluate yield. They evaluate abiotic stress resistance or tolerance, like cold damage. They evaluate plant architectural graft compatibility. And for each of these traits, you can use sensing techniques to be able to measure that. In addition to the crop traits or tree traits itself, they also evaluate the fruit quality. For example, of course, crispiness, juiciness are hard to evaluate using sensing techniques, but there are sensors that can measure the acidity. In terms of fruit appearance, many times they are just doing a visual rating. For example, you can see over here, they are rating from 1 to 5, but many of these can be done just using imaging techniques. So there are a lot of traits that are evaluated in standard breeding programs, and sensing can be applied in many of those. I also wanted to mention, if you really look at one of the big programs called as, this is a rose breed project, where they had a lot of crops that they were breeding for, mostly tree fruits, and they had established the standard principles of phenotyping. And you can see over here, one is commercial relevance, heritability and precision, availability of expertise, throughput, personal fitting. There are a number of criteria that you need to consider before you phenotype. And if you really see at least 5 of those 8 points, sensing methods can help do it much betterly. So for example, availability of expertise. Sensing is much more standardized that you do not need an expert to say whether this is light colored or dark colored, or this color is perfect from a consumer point of view. It is rapid once you establish your protocol. It's not subject to personal fatigue. It is cost effective, and it provides you a standard way of evaluating the phenotypes or traits. Okay. So with that, I want to start this example where we were studying the fire blight resistance in apple. Fire blight is an important trait of interest. Disease. It's an important disease. It's a common disease in Washington. So most of the varieties that are developed, they evaluate it for fire blight resistance. Okay, and it's a huge issue, and once it can just destroy the entire orchard. So it's of great importance to Washington state and other parts of the world as well. I would like to mention Wenatchee is called the apple capital of the world. So this is a very big, especially the central Washington region. I'm talking about Washington state. Many people think we are in Washington, DC. So Washington, DC is in the other side of the country. We are in Washington state, and we have a lot of apple production in this area. So the general the phenotyping procedure is they look at the shoot symptoms, which is in the bottom picture over here, and rate them from 0 to 3. That's the standard measurement or standard way of evaluating the disease resistance. But in our research, we evaluated a number of different techniques. So the first one, we used a hyperspectral sensor. This was a proximal sensor that was used. In the second aspect, we integrated the RGB and multispectral camera from a tractor or a sort of agricultural utility vehicle, and we used unmanned aerial systems to do multispectral based imaging. And you can see when you compare the visual rating, sometimes it matches and sometimes it not. I should say that if you're looking at one small shoot in the entire tree canopy, it is hard to differentiate. But overall, there was a slight trend where you can identify some changes in the canopy using some of this multispectral and RGB imaging. We also did a side imaging, and we wanted to identify. So you see that all that orange tagged branches are inoculated with the bacteria, and some of those were destroyed, and we could easily differentiate the dead or the length of those dead shoots in comparison to the background. So finally, with respect to the hyperspectral data, we applied partial least square regression as well as quadratic support vector machine. These are two types of machine learning techniques. We applied some of this, and we found that when the plants are young, the ability to differentiate some of these symptoms are much earlier and better than the mature leaves. So that was one of our conclusion, and we were able to publish this work as well. I would like to mention that we use the hyperspectral data to extract a feature called as normalized difference spectral index, which is a ratio of two wavelength combinations. So if there are 100, it is 100 permutation. So we could calculate a number of features, and we analyzed some of these as well using machine learning techniques and identified some of the ones which could give us a good indicator of visual rating. So this research was published in Frontiers of Plant Science, so feel free to read some of these published work as well. So I would like to summarize over here by saying that Phenomics is an integrated approach, though I'm talking mainly on sensing and application of sensing for evaluating some of the traits of phenotypes. But we closely work with the breeders. It's important for us to understand what we are trying to evaluate before we can apply some of these sensing techniques. So that's one aspect of it, where we need the engineers or data scientists need to understand the science, and also the scientist also needs to understand what is the capabilities of engineering techniques or data mining techniques. Many of these breeders have 10 plus years of experience. So just by looking at a symptom, they can say, okay, what is this disease, right? So sometimes machine learning or even sensing methods are limited to the data size. The more the data we are going to have, the better can be the outcome. So we need to sometimes incorporate a breeder's wisdom into some of these techniques as well. I would also like to say that visualization is very important. So just by analysis, if I do analysis and I'm going to give a breeder some complicated equation, the breeder is not going to use some of those. So they really need to understand, and probably if we can present the sensing data in a visualistic form that can assist them in making a decision, that's going to be very useful as well. Okay. With that, I just wanted to provide some of the overview of our ongoing project. I did mention that we are working on app development. We are transforming some of the sensing solutions into apps for better impact of those applications. We work on remote sensing, as well as we work on machine vision and developing image processing pipelines. Dr. Chongyuan Zhang, who will be speaking after me, will give you an overview on some of these pipelines. In addition to that, we apply machine learning and data analytics to understand the data better. We are also implementing IoT-based crop sensing technology, Internet of Things. These are low-cost Android-based sensors that we are placing them in field for continuous monitoring, as well as we are interested in biogenic volatile sensing. Though I talked mainly in terms of crop breeding, so you can use many of this sensing technique for stress detection, growth factors, produce quality, yield potential. The impact is not just on crop breeding, it can be applied to biology, agronomy, as well as precision agriculture application. With that, I would like to thank my students and postdocs. I have a very, very productive lab, both past and present. Without my lab personnel, it's hard to achieve many of the things that I stated. So I would like to give them all the credit. Most of the research was funded by USDA NIFA, which is US Department of Agriculture. So it is federal funding as well as state funding in terms of Washington State Department of Agriculture, Specialty Crop Lab Grant, as well as college support. And finally, I would like to thank all my colleagues. So these are different breeders, plant biologists, physiologists, whom I have closely worked with. And my contact information is as in this slide. Feel free to contact me. We are always looking for highly motivated students. So if you're one of those, please feel free to reach out. With that, I would like to end my talk, and I think we still have some time for questions.
Okay. Thank you very much, ma'am, for delivering a very enthusiastic and interesting lecture on use of proximal and remote sensing data in crop phenotyping to support breeding programs. So, ma'am, your lecture will definitely be more useful to the all the young students and young researchers who have joined this training program.
Thank you.
Okay. There are some questions in chat box, and some have raised hands. For the paucity of time, we may not able to take all the questions, but with your permission, I would like to take a few important questions.
Sure, sure.
Okay. First question, Dr. Pawan Kulwal from Biotechnology Department of MPQ Rauri has a question. Can UAV be used to measure phenotypic data if the population grown in the field is very diverse?
So most of the populations that we work are diversity panels. So sometimes they have diversity panels. I would say it depends on trait of interest. So for example, it's just plant height or traits like standard. So plant height is depend, even if it's diverse, it means the same thing. So I would say it depends on your trait of interest, and yes, some of these sensing techniques can be used for measuring.
Okay, thank you, ma'am. Then next, Mr. Balasaheb Dhame has one query. What will be percentage variation in reference accuracy on outer space prediction by using remote sensing and IoT?
Can you repeat that question again?
Okay,
Remote
okay,
sensing and
remote sensing and IoTs.
IoT. So spatially, the scale is very different. So if you look at satellite-based imagery, the best resolution we have so far is 30 centimeter per pixel. That is the best resolution we get. We have used this high resolution data in one of our recent work in maize plant, and we got very good, at least decent correlation that was comparable to unmanned aerial system. But when it comes to IoT, you're looking at millimeter scale. So unless if your plot is big and if your plot is uniform, there may be some relationship between an IoT sensor and satellite. But personally, I feel if you're able to put an IoT sensor in your field, I don't think so you need satellite-based imagery to do the same. So you have to pick and choose. And I feel if you can implement IoT, you do not need a satellite-based imagery for phenotyping application.
Okay, thank you, ma'am. Then Mr. Dhame has asked one more question. How remote sensing will help in yield estimation through the spikes analysis in rice and wheat?
So there have been a lot of studies, at least if you go to the just Google Scholar, there are a lot of literature, at least I cannot say a lot, at least there are a handful of literature where people have used unmanned aerial system or drone-based imagery for spike detection. And this spike detection, I'm specifically, I haven't searched for rice, but I have come across few publications on wheat spikes. So people are doing wheat spike detection using unmanned aerial systems. I would just encourage you find out some of those literature for better knowledge.
Okay, thank you, ma'am. And next, Miss S. Pavitra from Tamil Nadu Agriculture University. He asked, is there any possibilities to phenotype on when density using optical sensors over large population?
What density? Can you repeat that again? Sorry.
Is there any possibilities to phenotype on when density using optical sensors over large population?
Grain density, people have used load-based sensors. I don't think so. People have used basically optical sensing. People use optical sensing more for quality estimation, but not for grain density. So I would, there are sensors, but mainly load-based sensors, not optical sensing-based sensors for that.
Okay, thank you, ma'am. Next, Mr. Usman Abdullah from Nigeria has asked, what are the fruit quality components that can be assessed using a remote sensing technology?
fruit quality, I was mainly referring to fruit quality traits using proximal sensing. So nothing on remote sensing. You can only measure canopy based traits using remote sensing techniques. But if you want to evaluate fruit quality, you can measure color, you can measure shape, you can measure certain properties using near infrared based sensing as well. But most of these need to be in proximal sensing.
Okay, thank you, ma'am. Then next, Mr. Dibakar Ghosh from Bhubaneswar has one query. It is possible to differentiate a crop mimicry weed from that associated crop?
What mimicry you mean?
He asked, it is possible to differentiate crop mimicry weed from the associated crops?
Yes, it depends on your weed phenological stage of your crop as well as weed. So if your crop is masking the weed, it's hard to differentiate. But if there are certain different architectural traits of your weed with respect to your crops, then you can use remote sensing to differentiate those two.
Okay, thank you, ma'am. Next, Mr. Safal Rai from West Bengal has a question. What is the scope of remote sensing in hilly areas where we prefer contour planting for cultivation of crops?
Yeah, hilly regions are going to be much more challenging than the flat land. But nowadays, the drone-based technology has some advanced features. So, for example, they have one advanced feature called the follow the terrain. So if there is slope, your drone is going to fly in the same slope as your hill itself. But again, you need to apply image processing methods to normalize your data for those slope effects before you analyze it.
Okay, thank you, ma'am. Then next, Miss Aishwarya Gawande from Mahatma Phule Krishi Vidyapith Rauri has one query. Is there any technology to detect underground diseases or pests?
So most of our underground disease, for example, in our work, we have worked with Phenomics root rot, which is a type of root rot in lentils as well as pea. We mainly look at above ground symptoms. We do not look at underground symptoms basically. But there are people who put soil-based sensors, and volatiles could be another way of approaching the issues. But at least the breeders I work with closely look at above ground symptoms as an effect of underground diseases.
Okay, thank you, ma'am. Next, Engineer Tamboli sir or Dr. Vaibhav sir, any question from your side? Any important question?
There is no question today. You can continue.
Okay. Thank you very much, ma'am. There are still few questions and queries. We shall try to pass on those to the experts and convey you the response of experts through WhatsApp group or emails. So thank you very much, ma'am. Again, now
Yeah,
I would like.
Thank you very much.
Okay, thank you, ma'am.
Yeah.
Now I would like to request Dr. Mukund Shinde sir, organizing secretary of this training program, to propose vote of thanks and introduce our next speaker.
Yeah, okay, thanks. Thank you, Dr. Sindhuja. Very nice, informative lecture. And as you see, there are more than 700 participants for this training from more than 20 countries. Though most of the majority of them are from India. And if you see their profile, most of them are young actually. So I hope they will get motivated from your lecture. And as you appeal, maybe some bright students, they will approach you. And the lecture was very informative, very nice. On behalf of the Center for Advanced Agriculture Science and Technology, I thank you very much, ma'am. Thank you.
Thank you very much.
Okay. Thank you, Dr. Shinde sir. Now we are now start the second session of the day. And the expert is Dr. Chongyuan Zhang, postdoctoral research associate, biological system engineering, WSU. So, on the behalf of CAST project of Mahatma Phule Krishi Vidyapit Rahuri, I extend warm welcome to you, sir. And now I would like to request Dr. Mukund Shinde, sir, organizing secretary of this training program and co-principal investigator of CAST project, to introduce Dr. Zhang, sir, to our participants.
Yeah, thank you. Hello, Dr. Zhang. He is a PhD in agricultural automation engineering from of BSc at Washington State University in 2015 and worked on development of high throughput phenotyping technologies under supervision of Dr. Sindhuja Sankaran. More specifically, he applied high throughput phenotyping technologies to monitor Asocha blight disease severity in chickpea, detect flowering of 4 cool season crops, and estimate plant height of 3 cool season crops. To this end, he integrated passive and active sensors with different platforms, especially UAS collected data for crops using various platforms, for example, UAS and Tractor, developed image data processing algorithms like MATLAB and QGIS, and used statistical analysis and machine learning on extracted data for data mining. He is a licensed small UAS pilot and responsible for repairing and maintaining UAS and sensors. For example, configured and autopilot and cameras of Phenomics Lab. So I request Dr. Zhang to deliver your lecture. Dr. Zhang will be speaking on unmanned aerial systems, image acquisition protocol, and semi-automated plot-based analysis, its introduction and demo. Welcome, Dr. Zhang.
Okay, thank you. Can you hear me clearly?
Yeah.
Yes, sir. Yeah.
Okay. Now it's full screen. Is it from your side? Is it full screen?
Yes.
Yes.
Okay. Thank you for this opportunity to for me to present the image analyzing of unmanned aerial vehicle-based data for phenotyping application. Okay, here is the general procedure of hyperphenotyping use UAV. So usually we take the drone to the field, usually the breeding pod, and then acquire the image using the UAV and RGB camera or multi-spectrum camera. And then when we go to the lab, we analyze the data and extract the feature using either commercial software or customized image analyzing algorithm. And then we extract feature and compare with the ground truth. And when we analyze those data, we use statistic or machine learning. It depends on the study. And in this presentation, I will cover 3 major area: UAV-based data acquisition, UAV image processing, and UAV data analyzing. And then at the end, I will have a demo for the image processing. So UAV is getting very popular in the recent years, and here I've some reason or some of the advantages. For example, it's automatic, and it reduces the cost of labor. So when you go to the field and set the mission, and then you can fly the drone and then set it automatic, and it collect the image for you, and then you can cover a large area, and it depends on what kind of drone you use, and and also it's repeatable. So once the mission was created, and the next, for example, if 5 days later you go to the field and you upload the mission, and then you can send the drone to the skies and then acquire data again. So some of the software, so if you want to use a UAV for data acquisition, so you need some software for the mission planning. Some of the popular software is listed here. For example, mission planning is open source and free. And in our lab, we use this one. And also other software, including Pix4D Capture, DJI, and other commercial software such as Atlas, Fly, or Field Agent. The last two go with some of the multi-spectrum camera. So when you choose those software, some factor you need to consider, for example, the compatibility between the camera and UAV, and also the cost. Some of the software, I think one of these, you can have the basic version, but if you have some more automated function, you need to pay for the subscription. And after you make the mission and then you upload the mission to the drone, and then you are ready to fly it. So and after you start the program, the drone will go to the sky, fly automatically, as you can see in the slide. For example, at the lower right corner, showing the mission and showing the drone and carry out a mission. So where it complete the data acquisition, you will see a tiny camera, and also you can see some of the parameter of the drone. So those are very simple. Some general information about data acquisition and using the drone. And after the acquire the data, and we go back to the lab, we need to process the image. So first, here, all the image acquired is individual image, cover a small area. So we need to use some software to stitch the image and to create an image cover the entire field. So some of the popular software for the image stitching, including Agisoft and Pix4D Mapper. Those are commercial software. And another one is open source, is Open Drone Map. You can try this one in your computer, it's free. And for more information about image stitching, it will be presented by Montano tomorrow. So she worked for Pix4D. If you're interested and enjoy the presentation tomorrow. And here I'm not going to go too deep about those details. So after you put the image in the software, and then the software you choose some template to stitch the image, and then after 40 minutes, 1 hour, the software will give you some result. For example, this was one of the stitch image from my cousin Red Edge camera. It contains 34 image. And also, besides 2D image, those software you can create a 3D model of the field. So this one is can be used to estimate the plan height, as Dr. Sindhuja Sankaran mentioned before. So this is one of my PhD study. So we try to estimate the plan height. Okay. So after you get the stitch image, so you need to use some kind of software or algorithm to extract feature from. Sorry. So you need some kind of software to extract feature from individual pod, just like the cursor pointing. So you need to some kind of software to separate extract feature from each individual pod. So some of the feature we usually extract vegetation index, canopy area, canopy temperature, plant height, flower intensity, or extra. So vegetation index is a combination of refractant waveband of vegetation index is combination of refractant waveband of two or more waveband, which highlight the vegetation property. So I mean, in another word, it's an index to evaluate the healthiness of the vegetation. So some common vegetation index, including NDVI, GNDVI, and NDIE. NDVI stands for normalized difference vegetation index. It's a very simple formula and just require two waveband. You can search online for more of this detail. So here I will, for my study, I developed some image processing algorithm in MATLAB. MATLAB is a commercial software. One of the function is image processing. So, so we developed some algorithm to process our data. So here I use the vegetation index as an example. And you can refer to one of our publications for more detail about this algorithm. So first, after you got a stitch image and import into the algorithm, so first we need to rotate the image, so and which is to prepare for the port separation. So the software and the algorithm will rotate the image to align the port, to align the field in a to make sure the port is vertically or horizontally aligned. So it's to make the port separation in the following step is easier. And then so the algorithm prompts the user to input a degree to rotate the image. So the degree is, this one is a trial and error method. So the user can try multiple times and until he or she found a good degree. And then the next step is to crop the image. And so because image data is very big, especially after stitching. So it's sometimes it's a few megabyte or even some a few 1 or 2 gigabyte. So to speed up the image processing, it's better to crop the image. So in my algorithm, the following step is to select the field of inches. So just select the field and then the algorithm crop it. Following that, the algorithm automatically calculate different vegetation index or other features of inches. For example, plant height, and prepare those features for the next step. And this step is automatically, so this one no input is required. And next one, so we are interested in the canopy. So we don't want anything other than the canopy in the pot. So we exclude them by creating a mask. So a mask, so a mask is just a mask is to highlight the canopy we are interested in. So it's very simple. It just is to set a threshold in some of the vegetation index map. For example, here I use a soil adjusted vegetation index when I creating the mask. So I set a threshold to then create highlight the front ground, which is the canopy, and then and this step is also a try and error procedure. So you can inspect the result here, as you can see in the figure. I overlap the mask with the original image, so I can inspect the result. If it's not very good, so I can repeat this process and then create another mask. So after I got a good quality mask, and then I go to the next step. So we need to extract the feature from individual port, as you can see from the figure at the lower right corner. So we are interested in the individual port. So we need to separate the entire field in multiple ports. So here I think I have 88 something a port here is simple. So I can even draw manually, but sometimes we have up to 200 to 500 port. It's very tedious to do it manually. So I develop a procedure to identify the port semi-automatically. So what the algorithm means is the location information of the field. So in this step, the user need to identify the corner of the field, and with this information, the algorithm automatically separate the port, as you can see from the example here. And then after separating the port, region of interest will be created by removing the four edge of the port. So and then the feature will be only extracted within this region, and the soil was excluded. Only the canopy, the feature from the canopy was extracted. So the last step is to extract a feature from the individual from the region of interest within each port. So then the algorithm will automatically extract those features and then export it in Excel. So after data acquisition and image processing, and then the next step is to analyze the data. So the UAV-based feature, for example, canopy area, vegetation index, canopy temperature, will analyzing either using statistics or machine learning to correlate with some ground truth feature, or was used as a predictor to predict some of the ground truth data, for example, the yield of biomass or disease severity. Here I have two study case. So first is the disease severity monitor. So you can refer to our published paper for more information. So the goal of this study is to monitor the ascocartide blight disease severity in chickpea using remote sensing technology. So we collect multispectral and thermal image. For example, here is the thermal image of the field, and use those feature to predict the disease severity. So basically, the image-based feature most of the image-based feature are significantly correlate with the ground truth. For example, the visual weighting and the yield. And as you can see from the figure, and the correlation is high, very high after the disease symptom stabilize. And also the image-based feature was highly correlated with the yield until it reached mature. So Another study, in another study, I use remote sensing to monitor the flower intensity. And in the group, flower, the flower is related to the yield. So also, it's also especially in Washington State. So the plant need to flower and mature before the hot summer come. So here in the summer, basically we don't have rain, and so everything, all the plant need to complete their life cycle. So the right flowering at the flowering time is important for canola, pea, and so the breeder want some want to develop some cultivar that is mature early enough so it can finish the life cycle. So this the goal. So however, when you use visual weighting, they use visual weighting to evaluate the flower intensity for a long time. However, is the flower intensity is it's difficult to tell. The variation between pot is very minor. So if you want to give a very good estimate, it's a little bit challenging. So we try to use the camera and remote sensing to evaluate objectively and automatically. So in this study, we have a good correlation between the image-based feature and then the ground truth. And as you can see from the high R value, and you can see some of the resulting image. So this one, this image is from, I think it's from 15 meter. And as you can see, the algorithm can detect the flower accurately. So those three steps is the general procedure for the image. For the UAV based phenotyping. So here I'm going to introduce you for another image processing algorithm, because the previous one we introduced is not very. It's not well prepared and published to the published for Dano. So however, there's another one is already published, and then you can download if you're using R for statistic, and you can use this R package for image processing as well. So for more detail, more detail about this R package, you can refer to this paper. So currently, and this R package can analyze RGB, multispectrum, and hyperspectrum and crop surface model data. So it covers most of the data for phenotyping application. So it's very useful. So some of the function is is listed here. So as you can see, it's pretty close to the one I developed, and including selecting the field, rotate the image, calculate the vegetation index, and I did create a shapefile or separate the plot, and then calculate the feature, export the feature, and saving the file. So currently, this R package, or called field image R, can extract vegetation index, the number of objects. By the way, this R package can handle remote sensing image and some proximal image. For example, you take a photo of the ear of the corn, and you want to calculate the number of seed on the ear. You can use this package as well. And but I'm not going to cover those features here. also, this software can estimate the canopy area, plant height, and distance between plants. So yeah, this is a basic function about this software. So next, I will demonstrate very quick. So let me switch to the RStudio very quick. Okay. Studio. Okay, can you see it? RStudio?
Yes, yes.
Okay, thank you. So this one is free, so you can download from GitHub, or you can just download. You can download even in the RStudio. So before you use it, you need to download some package as list here. So some of the package, and then you need to load the library. And then, by the way, you can find more information about this software package in this website. So it covers both the free software for stitching for image stitching, the open drone map, and cover the view image R. So you can, if you're interested, you feel free to study over there. And then, okay. So first we need to, first load the image. Let's see. Okay. Yeah, okay, I hope you can see the image. So here's the one of the, yeah, this one is the field in the power intensity evaluation study. So this one is canola. So after loading the image, and then you need, rotate and crop the image first. Okay, so first crop the, oh, shit. Okay. Okay. Okay, after, yeah, so crop the image, and then next step is to rotate the image. So here, the image rotation is similar to the algorithm I developed. First, is try and error. So first, you need to identify the degree. So let's see here. Okay. So can I? Okay. Okay. So first, so it identify 2 points and create a light. So it will, okay, so when you create a vertical or horizontal light, and then it will calculate the degree, and then you need to put this the degree to rotate in the next step. Oh, sorry. Yeah, this one is some kind of trial and error step. So, okay. Yeah, and then original and the rotate image is so side by side because this image is not, I mean, it's almost ready, so it's difficult to tell the difference. Anyway, so next, after you rotate the image and prepare for the plot separation, the next step is to create a mask to separate the canopy. Okay, the mask is created, and then it will remove the soil and highlight the canopy. Next step, yeah, this one it will calculate a couple index. So the default index is can be listed here, which I highlighted, and you can also calculate some custom vegetation index. So I put one here, and just see the how well it works. And, okay, after yes, so after the index calculation, it will list all those index map, including individual channel and some of the vegetation index, and including the index, including the customer index. I put in the code, and then so here I also demonstrate calculate the canopy area. There's something wrong. Okay. Okay, I think I make some change here. So let's skip the canopy area. Okay, and then after you calculate all the index, all the feature you're interested in, for example, vegetation index, canopy area, and plant height, the next step is to identify, create, to separate the plot. What's wrong with this? Mm-hmm. Okay, there's something wrong. Sorry, I don't know what I changed and make. Just a second. Oh, yeah. Okay, just a sec. I think I blocked this. Okay. Yes, I blocked this. I blocked this. I forgot this this line of code. So now it's working. So the next step is to identify the field. So it's very similar to what I mentioned. And then identify the. So need to identify the field. Yeah, I didn't select. Oh, sorry, I didn't select the field properly. But anyway, so this the similar procedure as I mentioned before, and then so it always it separate the port as you can see here. So I mean, you can play around with this software here. Okay. So next step, after you extract the, prepare all the step, prepare the feature map, prepare the shape file, and then you will calculate all the feature, calculate all the feature, sorry, and then you will extract feature from individual port, and then export the feature using the function. So here I'm not going to run this code. It will take a while. So some comment about this, sorry, some suggestion about this software. So this one is better to use a desktop to run this software, and especially when the image is very big. For example, a few hundred gigabyte. So it will. So I'm running this software in my laptop. It's a little bit slow, so I suggesting using the desktop. And if you're interested in this software, please feel free to go to their website to learn more about this software. So pretty much I want to demonstrate today. So I'm glad to answer the question.
Okay. Thank you very much, sir, for delivering a very informative lecture on image analysis of UAV-based data for phenotyping application. This is very important topic, and your lecture will definitely be more useful to the participants who have joined this training program. There are some questions in chat box for the okay, but with your permission, I would like to take few important questions.
Oh,
Okay.
sure.
Okay. So first question, Dr. Sangan Basav Golagi from Bagalkot has a question. UAV can be used for protected cultivation.
Protective use for protective cultivation.
Okay. UAV can be used for protected cultivation.
So I'm not sure how he defined the protective cultivation. Can
Okay, sir. I will take the next question. Okay. Then next one, Dr. Tushar Patel from Navsari has one question. Any ongoing project regarding your topic? It's ongoing in India, or is there any collaboration with any Indian institute?
Not yet. We are working. We don't have any cooperation yet. But if you're interested, feel free to contact Dr. Love Court or Dr. Sindhuja Sankaran. You can talk with them for potential cooperation.
Okay, thank you, sir. Then next, Mr. Bhavya Pal has asked two questions. I take first of all, first question. Suppose a farmers need to fly the drone in their farm for any kind of survey. So he or she need any kind of permission to fly the UAV.
This is a good question. I'm not sure in India, but in US, it's required to have a license. So it is called small UAV pilot license. So it different from country to country. So you need to check the local government.
Okay, thank you, sir. He asked one more question. It is possible for a small scale farmer to buy this kind of UAV So if they need this drone, so from where they get kind of subsidies to get the drones.
आहे का साहेब तर
Sorry.
Can I repeat?
Yeah, sure.
Okay. It is possible for small scale farmers to buy this kind of UAV. So if they need this drone, so from where they get some kind of subsidies to get the drones.
For the subsidy, you need to contact the local government, but you can buy some low-cost UAV. For example, some of the custom-grade UAV can be used for phenotyping purpose. Actually, if you remember in my slide, we have the Y UAV is a custom-grade UAV with RGB camera. It's low cost. It can use for multi-purpose. For example, I use that one for foul detection, plant height estimation, and you can even use for DC. So I think it's possible to buy a low-cost UAV for that purpose.
Okay, thank you, sir. Then next, Mr. Golmai from Raipur has one query. He asked for calculating the plant height. Can we measure only the selected plant or simply sample only from each plot?
Okay, thank you. That's a good question. So for us, when we're using the remote sensing, we measure entire plot. For the breeder, they select a few plants. So this one, this is one of the advantage of using remote sensing compared to the manual method.
Okay, thank you, sir. Then next, Dr. Mangesh Badguzar from Nasik, Maharashtra has one question. He asked, can we measure the total plant population per hectare at the time of harvest in densely populated crops like sugarcane, paddy?
I'm not sure what feature he or she interesting, but for the remote sensing, yes, that we can using UAV to map the entire field. One per. A few acre. You can use the multi-copter or fixed wing. Fixed wing can cover more area, so I think it's possible. But it depends on what kind of feature he or she interested in.
Okay, thank you, sir. The next, Mr. Siddharth has a question. Is all the images sensing software are calibrated with pilot testing prior to actual use?
Sorry.
Okay, I will repeat. Is all these image sensing software are calibrated with pilot testing prior to actual use?
It depends on. It may be need to modify some of the parameter slightly because, for example, when creating the mask, the threshold may be different from crop to crop, so you need to change this slightly. And also it depends on the feature you're interested in. For example, flowering, it depends on what kind of threshold you're setting.
Okay, thank you, sir. Then next, Mr. Manish Bedar has one query. Is there UAE technology can provide sustainable solution for agriculture farming?
So is any UAV technology can provide sustainable solution for the
Agriculture farming.
UAV it can help. UAV can help solve some of the problem. For example, monitor the disease, lodging or something. But you need to other technology to help. Combination, not just. UAV technology cannot solve all the problem.
Okay. Then thank you, sir. The next, Mr. Gopikar has one question. Can we use UAV technology in crop cultivating in polyhouse? If yes, how can we take images inside the polyhouse?
Sorry, what's the inside?
I will repeat.
Inside what?
Can we use UAV technology in crop cultivating in polyhouses? And if yes, how can we take images inside the polyhouse?
Polyhouse. You mean greenhouse?
Yeah. He is asking about greenhouses only because protector cultivation in India, it is a greenhouse. They are talking about polyhouses. So it is.
Okay, so polyhouse, it means greenhouse. Yeah, I mean, can we use UAV in crops?
Actually, okay.
So UAV, I mean, UAV is more designed for field. So I mean, when you have some structure, it maybe have some interference to the GPS signal or something. It's possible, but not suggest.
I think.
Dr. Nanavade, I will answer this question.
Yeah.
This is regarding UAV, that is drone technology in crop cultivating in polyhouse actually. In fact, I will rate this as very good question because UAV still recently were not thought to be used in polyhouse. Okay, but so we are discussing with some experts and what we call the growers in polyhouse who adopt the practices of hydroponics and aeroponics. Okay, please underline hydroponics and aeroponics. Okay, so in that case, the need was realized. Okay, to identify the pest and disease, okay, of the individual fruit, say capsicum. Okay, so in that case, then we came out with the plan, which of course we have not started yet executing. That if we have the UAV, small UAV, okay, mounted with one multispectral sensors actually, and if we keep on flying that small UAV with multispectral sensor, that UAV will capture the images, and with the help of AI, we can say that whether the infection has started or not actually. Okay, so in case of the hydroponics with polyhouse, it is necessary because if there is only identified for say few hours, then it is quite possible that whole crop in the polyhouse will be damaged actually. Okay, so for that the technology was needed, and the solution was that the UAV mounted sensor, okay, so which will pick up the infestation, okay, of the individual fruit, and then we can identify that individual fruit and we can treat that actually. So it is possible, and the thoughts are actually already going on. And if any one of you, if you are interested, so you can join our research program. Okay, so the details are provided in COST website actually. Thank you very much for this question.
Okay, thank you, sir. Then next one, Ruthir Jungle from Ratnagiri has one question. What are the different paths used in UAV to scan a field?
Okay, thank you. So we usually call it if you remember in one of my slide, so it's line by line. Each line is parallel. We call this one is single grid. It's just like a zigzag pattern. So it's one file. So but if we want to create to estimate the plant height, we usually use the double grid. So it's very simple. We fly, and for example, the first fly is north and south. The second fly is east west. So it's two fly, and on the same field to cover more. We call this one double grid. Usually, is these two methods for the automated data acquisition.
Okay, thank you, sir. Then next, Miss Himasweta from Bhubaneswar has one query. He asked, what is the minimum cost of UAV?
Minimum, I mean, some of them, some custom, I think some of them maybe, if it's created by some commercial UAV company, for example, DJI, I think some of them is $1,000 US, but some of them you can make it, some of the UAV development kit, you can make it, I think, $300 or $400 US probably. So I'm not doing a good. I'm not studying very well, but 300 or 400 may be possible for RGB based and simple UAV.
Nandalal, most of these questions are related to Indian conditions. So kindly go through those questions which are related to expert. Other questions we will be answering afterwards, or we can give those answers in our concluding session or with the mail. Please take those questions only which are concerned with the expert and related to this lecture. Because these are general queries and we can answer these queries afterwards.
Okay, okay, okay. Okay. Engineer Tambori sir, any question from important question from your side?
Yes, sir. I will ask one question. Mr. Jitendra Singh, he has asked that how to use MATLAB for calculation of water requirement.
Use MATLAB for calculate water requirement. So
of
usually,
any crop.
Oh yeah. So thank you. So usually, in one of the study, as Sindhuja Sankaran mentioned, so we estimate the performance. So and the performance of a potato or grape. So we evaluate the treatment. So for example, 30% of water, 60% of water, and so it will we evaluate the performance and then see if it's possible to reduce the water, or and in another way, we monitor if the plant is under stress. For example, this the plant is under stress, so we need to water them. Is another method.
Okay, sir. Thank you very much. One more question. Mr. Abdullah from Nigeria, he has asked that, can you tell something about role of big data in agricultural domain?
Big data.
Role of big data technology or big data analytics.
Okay. Yeah. So you know, the data, I mean, in agriculture, I mean, the data is huge and very diverse. So we use machine learning and for data mining, and we try to predict the performance of the plant and also predict the yield or something like that. So it's, I mean, we try to exploit those machine learning, data mining. So I think maybe helpful. Yeah, I'm not sure, but maybe that's the future.
Thank you, sir. Dr. Nandalal, you can continue.
Yes, sir. Thank you, Engineer Tambori. Thank you very much, Dr. Zhang sir, for replying and answering all the questions asked by the participants. For the information of all the participants, there are still few questions and queries in chat box. We shall try to pass on those to the experts and convey you the responses of experts through WhatsApp group or email. Okay.
Yeah. Excuse me, Deshmukh. Yeah.
Yes, sir.
I have one important announcement. Tomorrow morning, the first lecture is at 10 o'clock. And after that lecture, there will be brief presentation on real-time irrigation water management by Dr. Sunil Gorantiwar, Principal Investigator of the Center for Advanced Agricultural Science and Technology. on climate smart agriculture and water management, MPKV Rauri. I request participants to take the advantage of this presentation because we got this from the feedback of questions from the participants that the majority of them are interested in this water management. So definitely it will be useful to you. Thank you.
Okay. Thank you, Dr. Sinde sir, for announcement. Now I would like to request Dr. Sachin Nalode sir, organizing secretary of this training program, to propose vote of thanks.
Thank you, Nandalal. And it is a very good lecture and actual hands-on training he has given by the Chongyuan Zhang, who is working late night from US. I hope most of the questions which were asked, those were really not related to your work. So it was difficult to answer those queries because protected cultivation, we can have that UAV, which is cheated UAV. Actually, the studies are going on and we can work on it. Small, very small, that is nano-sized drones are usually used. You can go for UGVs rather than UAVs there. So it is rather another thing, and costing, etc. We can have another training session afterwards. Those who are interested, they can participate. So from my behalf and the team cast, that is Center for Advanced Science and Technology, MPKV Rahuri and India, we are very much thankful to Chongyuan and for his presentation, his queries and support to this training. Thank you very much. Also, thanks to our PI, Dr. Gorantiwar, who given a few answers to your queries, and he is ready to give answers to one of the query for calculating irrigation requirement and related softwares. So not only MATLAB, how to use other softwares and what are the relative terminologies and sensors he will discuss in tomorrow's lecture. Thank you, sir. Also, I will thank to organizing secretary Dr. Shinde and our team, Dr. Nandalal, Mohsin, Yogesh, and Vaibhav, who are managing this training day and night. They will be giving you assignments also and getting the assignment checked. So thank you very much for joining this session, and tomorrow's session will be there in morning also and in afternoon also. So you are requested to join those sessions exactly at the time. Thank you, Chongyong Zhang.
Thank you
Mr.
for
Zhang
inviting.
is there? Oh, sir.
Thank you for
Yeah, thank
inviting
you
me.
very much for your. Yeah.
Okay.
So,
Thank you.
Okay. Thank you, Dr. Nalode sir. Before we end the session, we have few announcements. In spite of our repeated appeals and guidelines, some of the participants have not joined by their names followed by name of organizations to which they belong. So this is important for recording your attendance during the sessions, to recognize the registered participants and also for security purpose. So if found tomorrow not joined by their names followed by organizations will be removed. They have, however, the option of joining through YouTube. Then, as informed in the beginning, we shall post one short assignment on WhatsApp group at 4 p.m. Indian Standard Time, and all participants are requested to submit the assignment on time and as per the guideline issued on WhatsApp group. So please note that responding to this assignment will be considered for issuing the certificates to the participants. So tomorrow there are two sessions. First session will start at 9 a.m. Indian Standard Time, and for that session, Miss Momtanu Chakravarti, PIX 4D North America, will delivering the lecture on decoding drone imagery with PIX 4D. And second session will start at 3 p.m. Indian Standard Time. For that session, Dr. Harshal Galgale, Senior Software Engineer, Kisan Hub, Cambridge, UK, will delivering the lecture on remote monitoring in agriculture. So all participants are requested to join tomorrow morning at 15 minutes prior to schedule time. So thank you very much. I ask to co-convener Dr. Sunil Gorantia, sir. Shall we close the session, sir?
Yes, you can close.
Okay, sir. So with the permission of from the co-convener and organizing secretary of this training program, we would like to close the session. See you tomorrow at 45 to 45 a.m.